electroporation technique using the Axoporator 800A from Axon Instruments for labeling and transfecting brain neurons for in vivo imaging and study. Now, let's start today's presentation. So now I will hand the ball to our speaker, Kurt. Here you go, Dr. Kurt.
Thank you, Jeffrey.
Yep.
Okay, so today I'll be discussing our use of the Axoporator 800A for transfection and dye labeling of individual neurons within an in vivo system. And the topics I'll go over today are the basics of electroporation, the introduction to single-cell electroporation, or SCE, explaining why we use this and generally how to set this up perhaps in your own lab, and how the Axoporator fits into this setup. And I'll talk about single-cell electroporation of small molecules versus DNA for transfection. And we'll also discuss the difference between blind SCE versus targeted SCE, and how we use this in our lab to look at in vivo imaging of neuronal growth. At the end, we'll have a session talking about troubleshooting of common problems. So what is electroporation in general? Well, it's a very old technique, originally used for transferring DNA in small molecules into cells within dissociated cultures or media. And that's sort of diagrammed here. We have a cell within a cuvette. There are electrodes on either side and a solution containing plasmid DNA. When we apply a voltage between the electrodes, the charged particles within the solution and within inside cells migrate towards the poles they're attracted to. So in this case, DNA is negative, it's going towards the positive pole, but within inside the cell, the ions also move, and this builds up a transmembrane potential at the membrane. So when we look at what's happening at the membrane, when we apply a voltage, there's an increase in the capacitance, the charge buildup. And when that charge, that transmembrane potential, reaches a threshold of about half a volt, the electrostatic forces holding the lipid bilayer together break down, and there's a reconfiguration of the lipids to form pores. And once these pores are formed, their lifetime is independent of the applied voltage, and they have lifetimes on the order of tens to hundreds of milliseconds. So while they're open, if the voltage is still applied, charged molecules, including plasmid DNA, shown here, enter the cell, and then once the voltage is turned off, the pores can reseal, trapping the compounds inside. So that's the standard electroporation technique. So back at the end of the 1990s and the early 2000s, electroporation was being applied towards living tissues. This was done in the chick spinal cord and retina. And in my lab, where I was working as a postdoc in Holly Klein's lab in the Coltsman Harbor Laboratory, we are applying it for transfecting large numbers of neurons in the tadpole brain by injecting DNA into the brain ventricle and applying external electrodes across the brain to transfect many cells. But our interest was to image individual cells, so we considered how to apply this technology towards single-cell approaches. And since my background is as a patch clamp electrophysiologist, the idea of approaching cells with a pipette was very straightforward. So we developed a technique called single-cell electroporation. This was done in collaboration with other people in the lab at the time, including Wun-Che Sin, Ashkan Javaharian, and Zheng Li. And it was a rather simple approach of restricting the electric field and the delivery compounds to an individual cell at the tip of a pipette. And this is diagrammed here with a pipette filled with a solution of DNA. There's a silver wire in the pipette and a ground wire. When you apply a voltage between them, a field generates at the tip of the pipette, electroporating only the cell that's contacted, and whatever is charged in the pipette is driven as a current into that cell through those pores. You turn off the voltage, remove the pipette, and in this case, DNA will be transcribed into RNA and translate into protein, perhaps expressing GFP. So this proved to be a very useful technique for labeling individual neurons, even in vivo preparation. In our case, the the Xenobus laevis tadpole brain. So what are the advantages of single-cell electroporation for labeling neurons? Certainly there are other strategies out there. There's viral transfection, there's transgenic techniques, biolistic gene guns, and liposuction are the most common. And they all have their pros and cons, their advantages and disadvantages. Certainly transgenics are very powerful, but you're restricted to genetic organisms, and they're very expensive and time-consuming. Virus is very powerful as well, but there's certainly species and cell type specificity, and there's time and cost associated with generating a virus. Gene gun biolistics is very useful, but typically restricted to cell culture preparations. And liposuction has variable ability to label individual cells and has residual delivery compounds. So for electroporation, the advantages are that it can be applied for both in vitro and in vivo preparations. You can transfect neurons at a post-differentiation state, which limits the amount of compensation that you see or may see with transgenic approaches. You can also apply electroporation to all organisms, even non-genetic organisms. So it really allows you to apply a real power of genetics to non-genetic systems. And you can use readily available expression plasmids. It's very easy to make these plasmids, and they're commonly available. And you can just go right ahead and deliver them to cells. You can also co-deliver multiple cells, genes at the same time on independent plasmids. There's a very high rate of co-delivery at the same time. In addition, the technique is very versatile for delivery of other compounds, not just DNA. You can deliver small molecules, including RNA, dyes, peptides, antisense RNA strategies like morpholinos and shRNA, drugs, and others. So anything that's charged can be delivered in this manner. And importantly, there's no residual agents. There's no viral particles, no gene gun bialistic particles, things like that. It's very powerful for having cell autonomous studies by transfecting or delivering compounds to an individual cell within an intact circuit. You're sure that the results that you see are due to the effects on that cell only, not secondary effects due to changes in the circuit. And it's very easy to set up single-cell electroporation, and I hope I'll be able to show you that. The downsides, there's always downsides for every technique, and we always worry that there may be damage. We are using a pipette that has to be inserted into tissue, and you always worry about the damage associated with that insertion, and you're applying electrical stimuli. That stimulation will induce pores, and it also can directly kill cells if the voltage or current is too high. The pores themselves, if they're too large, they won't reseal when you turn the voltage off, and they'll continue to grow and lyse the cell. So care has to be given to the stimulation parameters that you use. In all cases, you're creating water-permeable pores, so these cells will experience some osmotic shock and they will swell, but in our hands we see no apparent lasting damage from this. So the basic setup is very straightforward, just a microscope, manipulators, electrode holders, glass pipettes, along with a puller for those pipettes, and a stimulator. And today we'll be talking about the Axoporator 800A for the setup. There are two basic setups for single-cell electroporation. There's the blind SEE and targeted. In blind, you're inserting a pipette into a region where you know the cell bodies are, but you're not directly looking at the tip cell interactions. So this allows for a low magnification, long working distance microscopes, such as a dissecting microscope, and the use of coarse manipulators. This is very easy and cheap to set up, but you do have some limitations of efficiency if the lack of efficiency is due to lack of tip cell interactions. Targeted single-cell electroporation uses high magnification microscopes, such as a 2-photon microscope, to directly see the tip cell interactions. This greatly enhances the efficiency of electroporation, but it certainly takes more time to set up. It requires fine manipulators, usually motorized manipulators. So it's a sort of trade-off between the speed of doing multiple stimuli, what you get from the blind single cell, or reduced number but higher efficiency with targeted. For the stimuli that are used for electroporation, we often think of there being two different components to the stimuli. There's a pore formation period and electrophoretic transfer component. For the pore formation, we just need short duration, high voltage pulses to induce pores. And again, their lifetime is independent of the pulse after they're formed. And then you have a long duration, low voltage stimuli that's sufficient for electrophoretic transfer. So optimal pulse shapes might be seen as a biphasic pulse with initial high amplitude short duration pulse followed by a long duration low voltage pulse. Exponential decay pulses can mimic that biphasic pulse to some extent, but often people just use square pulses. And the worry there is that you might be inducing more damage. Alternatively, it's been found that trains of short pulses can provide efficient transfer without causing damage, and that's pretty much what we use in our lab. We either use trains of short pulses for DNA, or for small molecules, we either use just a single square pulse or a shorter train of short pulses. And we typically use the Axoporator 800A in our lab. We have other stimulators, but this is definitely our favorite. And it's really useful because it's designed for single-cell electroporation. It's the only stimulator out there that is. And a lot of its features are designed optimally for this process. So it allows you to select different types of pulse shapes, including biphasic pulses, square pulses. You can easily change the polarity. And you can drive either single pulses or trains. You're able to control the pulse width as well as the train frequency and, importantly, the train duration. Many stimulators don't have that type of capacity, and we find that's the most useful parameter that we change quite a bit. As well as it has the ability to present the tip resistance continuously. And we're constantly changing the voltage to compensate for slight amounts of clogging of our tips. So that's an incredibly useful feature. It also has a foot pedal trigger as well as a manual trigger, which is very convenient. So it's definitely our favorite stimulator in the lab. Okay, so another important aspect of single-cell electroporation for success are the tips. And I think this is where many people have problems. We find that the success is very sensitive to both the tip diameter and to the shank angle. In our hands, we find that a 0.6 to 1 micron tip diameter and rather shallow shanks are optimal. We find that there's a relationship between both that relates to the field strength at the tip of the pipette, and that field strength diminishes very rapidly as you move from the pipette tip. So all these features relate to whether you get single cells or multiple cells or no cells electroporated. If the tip is too small, typically it will clog, or the field strength will be too small to electroporate cells. As you get larger, you typically get single cells, and if you have a larger tip, you will get multiple cells labeled, and then larger tips will not have a sufficient field strength to generate electroporation. In general, we care about how much damage we induce by our tips. So very wide tips will produce more damage if you're pushing the tip deep into tissues. On the other hand, a thinner tip is more susceptible to breaking and clogging. So it's fortuitously in our system, in the tadpole, our cells that we're targeting are very closely localized underneath the skin, so a wider shank does not really create much damage in our system, but it's something to consider. But this is something that one should take a lot of effort to optimize tip geometry. And I'll talk about using dyes later on to work with tip geometry to optimize your parameters. So first I'll describe blind single-cell electroporation, and I'll describe how we use this in the tadpole brain. This is the Xenopus laelis tadpole. These organisms are transparent because they're albino, so we can directly image into the intact brain. That's the outline of the brain and the spinal cord. And this is the tadpole under a dissecting microscope showing the general setup. The tadpole's on a moistened Kim wipe. You can see there's a pipette inserted into the brain. The tadpole's anesthetized. In that pipette, there's a small amount of solution. This is DNA solution, and there's a wire inserted into the pipette touching that solution. You can see that we don't need very much solution in the tip, and this is useful even though we do reuse the same tip over and over again for multiple preparations for the same day, as long as it doesn't clog or break. It's nice not wasting a lot of solution in your tips if you don't need to. On the bottom left, you can see a ground wire that's in electrotonic contact with this tadpole, and we stimulate between those to electroporate cells. Here's a higher
Dr. Kurt,
yes?
Excuse me. So in your screen right now, I still see the Axoporator 808, the screen, right? Is the slides moving? Just double check.
Mine are moving.
But
okay,
I still see the Axoporator 808, the setting, the pose shape, right?
Okay, let me try to restart my screen.
Yep.
Do you see something different now?
No, still Axoporator 808 slides, right?
Okay.
Oh, yep. Right. Okay.
Is it changed now?
Yeah. I see the change right now. Right.
Okay. So let's say blind single cell.
Yeah. Can you go back up slide, previous slides?
Okay. This is the tip geometry slide.
Okay. Previous slides. Right. Right.
And
so I think I think you stopped at the slide that showed the Axoporator 808 screen, right? Right here,
okay,
right.
Can you see that now?
Yep, I can see it right now, right.
Okay. So I described this setup. This is a slide showing our tip geometries. You can see this is slide advancement, yes?
Yes. Thank you.
Great, thank you. So
Sorry for the interruption.
Oh, thank you. So this is the tip geometries. And now talking about the blind single cell electroporation, this is the introduction to our tadpoles, showing a transparent tadpoles and an outline of the brain and the spinal cord. These are very useful animals for studying brain circuit development because they offer a window into a developing brain in a transparent organism that we can immobilize while they're awake and directly image into their brains. And we use this organism by labeling cells in the brain using single cell electroporation. This is the image of the tadpole under a dissecting microscope showing the pipette inserted into the brain. Again, this pipette is filled with a small amount of DNA solution, and there's a wire in that pipette, and there's a ground wire on the bottom left, which we stimulate between these two sites. The next is a higher magnification of the tadpole's brain, showing the pipette inserted into the brain. We don't see the tip. Again, this is a blind technique. We know generally where the cell bodies are, so we just insert and stimulate, then move to another site, and usually hit about 5 or 6 sites per tadpole. Again, the efficiency may be limited based on whether the tip is in contact with a cell, but we can easily move between sites because we have a lot of easy working distance. And after waiting 24 hours after delivering DNA, this is a GFP expressing cell within the tadpole's brain. And this is that cell within the tadpole's entire head. So this is, you can really see why this is a useful technique for looking at neuronal morphology since only single cells are imaged. Next, this is showing a blind single cell electroporation of a dye. So you can see how quickly we move this pipette from site to site. We can go just over a period of seconds between different sites, and we just delivered fluorescent dye into cells. That's one cell labeled after a few attempts. Using dye obviously is very useful because you have immediate feedback of your single cell electroporation success that you don't get with DNA since you have to wait 24 hours. So dye, use of dye is very useful for getting immediate feedback to help optimize both your tip geometries and your stimulation protocols. This is where using something like the Axoporator 800 is very useful because you can rapidly change a number of parameters and have good control over things like the train duration. So next I'll show you what we do with this. We do in vivo 2-photon 3D imaging of our cells. This is a living tadpole brain cell. imaged in an awake tadpole, and we developed software for doing morphometrics to measure the axon and dendrites of these cells. And we do time-lapse imaging of neuronal morphology with focus on the elaboration of the dendritic arbor. This is a single tadpole cell imaged over 4 days, just taking snapshots of their structure to look at patterning of dendritic arbor growth. By doing rapid imaging of individual neurons in the awake brain, we get movies like this. Here, the cell is an actively growing cell. The portion here is the axon that's being cut off. On the left is the entire dendritic arbor. And by capturing imaging every few minutes, this whole movie is 3 hours, it's looping, we can capture all the rapid growth dynamics of a cell. And this is a really powerful way of looking at growth and using single-cell electroporation both for the morphology in control cells or by applying other genes to up or down regulate gene expression for studies of protein function. In my lab, we developed software to identify, track, and measure all processes in 4 dimensions. And that software we call Dynamo. And this is shown here. We have the same cell image at 5-minute intervals at the top, and it's overlaid on the top right. Our software, each time point is a full 3D stack through the dendritic arbor. Our software identifies, tracks, and measures every single process, providing length measures at the bottom that can be translated into motility and addition and retraction rates that are comprehensive. So this is a very rich data set for comprehensive quantification of dynamic growth behavior of living neurons in intact and awake brains. So for targeted single-cell electroporation, we also use this, and it's a very powerful technique for increasing efficiency, but also selecting the very neurons that you want to electroporate. So for here, we're using targeted single-cell electroporation of neurons based on their receptive field properties or their sensory response to external stimuli. And it's a 2-step process where we first load the brain with a calcium-sensitive dye, in this case OGB, Oregon Green Bapta. This is an AM ester dye, so it's taken up by cells and then cleaved internally by internal esterases, so it traps the dye inside the cells. After loading the dye into the brain, we mount the tadpole that's awake on a 2-photon microscope, and we provide visual stimuli. We can then find the cells that are the most responsive to the stimuli we're interested in, and then target those very cells for electroporation, in this case with a space-filling dye, Alexa 594 dextran. And we're using a short train of pulses to fill that cell that you can see in the top right. So in the bottom left is the calcium response in the cell body of this cell in response to visual stimulation, corresponding to its firing of action potentials. On the right is the filling, the 3D volume of the cell. So we can go back and forth between calcium imaging of its soma and its full 3D morphology to compare functional and structural plasticity. And this is an example of the structural plasticity of this cell. Here in the bottom left is a drawing of the cell using our Dynamo software. On the top right is an overlay of 2 time points, 0 and 30 minutes. And the bottom is a description of the growth behavior over that time point using our color scheme, where every process or small process or filopodia has a circle that's color-coded where green branches are newly added, red processes are subtracted, blue and purple processes are present at both the 0 and 30-minute time points. Blue ones are extending, purple retracting, and the diameter of the circle corresponds to the length of morphology changed over time. And from this, we get these 3-dimensional images of growth behaviors over time. Here, the gray circle is the cell body, and all the other circles represent how much morphological change there is and what type of change happens over time. Meanwhile, but again, we still are sampling the calcium transients within the cell body of these cells to look at both their functional responses and their structural changes. And all this is possible from single-cell electroporation. So when we think about what are the optimal stimulation parameters for generating these cells, The parameter space is really wide. There are many different types of parameters that you can adjust. And we've tried many different things from square pulses to exponential decay pulses to trains of pulses. And basically we find that while many different stimulation parameters work, there's definitely a voltage dependence. Probably at higher voltages we're killing cells. And we find that trains of high frequency pulses or square pulses works optimally. And, as we discussed before, is there any damage from these cells? And I think this is something you always have to ask with any of your manipulations. And the best that we can do to look at this is to look at how cells grow, record their electrophysiological properties, and look for any changes. And when we look at growth of cells labeled by a single cell electroporation, here's a cell that we've imaged every few hours and even days. We don't see any morphological phenotypes of damage. There's no blebbing, and they grow in manners that's very similar to other cells labeled by other means, such as dye-eye labeling. So we think this is a very sensitive measure, and we don't see any change, and we don't see any change of cells that are their growth behaviors if they're electroporated after 30 minutes or 24 hours later. We also don't see any changes in their electrophysiological properties. So we haven't detected any problems, but it's something you'd always want to keep in mind. So single cell of plasmid DNA is very different from delivery of small molecules. And I'll discuss the plasmid DNA first. So it has great strengths, especially for just looking at cell morphology with a space-filling fluorophore. If you deliver dyes into cells, they will bleach over time. If you deliver plasmids encoding things like GFP, they'll get brighter and brighter over time. And even if some of the fluorophore bleaches, it's replaced by other proteins. Obviously, you can control gene expression, and you can, as we discussed, you can transfect neurons different stages post-differentiate at different stages after they differentiate. So that limits the amount of compensation that occurs. You can apply combinations of plasmids at the same time. One can be a space filler, while other plasmids may encode proteins that are either fluorescently tagged to look at protein localization, or you can do protein studies using dominant negatives or constitutively active constructs. There are challenges with DNA. DNA likely has to get to the nucleus during the electroporation process to be effective, and that's the challenge. DNA is relatively large, so it requires longer trains of pulses than you would need for small molecule electroporation. These are the parameters that we've worked out for DNA for our cells. We do use trains of short square pulses, about 1 millisecond duration square pulses. We use a range of frequencies between 200 and 700 hertz, and train durations of 300 milliseconds. Our tip resistances are about 30 mega ohms, and we use 30 volts from the Axoporator. We use plasma DNA that's endotoxin-free, made with maxi preps, and we use concentrations around 0.5 to 2 microgram per microliter. And we only use, typically use this DNA dissolved in distilled water. We are electroporating the solution, so if we have other ions in our solution, we're going to be driving those ions as a current into cells. We don't see any difference in the efficiency with different types of salt solutions, so we simply use water at this point. The ability to co-electroporate different plasmids is very powerful, and with single cell electroporation, the co-electroporation efficiency is near 100%, and this is a case of delivering plasmids encoding GFP and DS-RED together, and we always see co-localization. This is a case where we're using expression of two different plasmids. One is to express GFP as a space filler, along with plasmid encoding a kinase, protein PK and zeta, to look at effects of overexpression of this protein. At the top is the control cell. It shows an overlay of 6 time points at 5-minute intervals, where the white shows the stable regions of the dendrite. The color shows places of dynamic motility. In the bottom, when we overexpress PK and zeta, we find that there's very little of this color, so it shows that these cells are frozen. They're not very motile. It's showing a role for PKM zeta in stabilizing dendrites. So this is an example of a cell autonomous experiment. Only this one cell is overexpressing this kinase, so we know the effects are due to the effects from this cell, not due to secondary changes from the circuit. This is a case where we're co-electroporating a red space-filling fluorophore, DS-red, along with a synaptic protein, PSD95, fused to GFP, so we can mark postsynaptic sites. In this example of us using single-cell electroporation to transfect rat hippocampal pyramidal neurons. These are cells in organotypic slice cultures. So just to show that you can use single-cell electroporation for other organisms besides tadpoles. However, we should note that this is more challenging, so I'd be a little cautious in working with DNA. So rather as opposed to DNA, electroporation of small molecules is very easy. And I think this is something that should be tried for any cell and any organism. Because you just have to transmit these compounds across the plasma membrane, which is very easy to do. You can use this for dyes, for space filling. You can apply peptides that are fluorescently tagged to label intercellular proteins. You can apply inhibitory peptides, RNAs, short hairpin RNAs, and also morpholinos, these synthetic antisense RNA constructs. You can also apply drugs or calcium-sensitive dyes, as well as many other compounds, as long as they're charged. The parameters for electroporation of dyes, in our lab, we use square pulses or short trains of pulses that are only about 15 milliseconds long. Again, the tip resistance is about 30 megohms, and we use a voltage about around 30 volts, but that varies depending on the tip. A common dye would be Electrofluor 48 dextran. Concentration range about 100 micromolar to 1 millimolar. Again, dissolved just in water. All these parameters we adjust for each tip. It's very easy to see the effect of electroporation using fluorescent imaging. However, you always have to be careful not to overfluoresce your cells or you'll kill them. So we're very conscious of that. And this is an example of co-electroporation, using single-cell electroporation of morpholinos with a fluorescent space-filling dye. We always fluorescently tag our morpholinos so we know that they're being delivered while we're filling the cell with a space-filling dye. So the morpholino is labeled in green. You always worry that when you use combinations of compounds that they might interfere with their delivery. So using fluorescently tagged compounds is very useful, whether that's morpholinos or peptides. This is a case where we are both electroporating a calcium indicator, OGB, along with a peptide that binds to PSD95 that's fused to a very bright squarene or taxine dye. So this is a case where we're using it for calcium imaging along with inserting a peptide to look for protein localization. So when we think about delivering compounds, you really have to worry about the polarity of that compound. And it's important to empirically test that under different conditions. Many cases occur where we think we know what the polarity of a molecule is, but either it acts its effective polarity is different from that, or over time it may even change. There are cases where a dye may move as one polarity after a number of trains. It will change its polarity probably due to ion change from the extracellular solution, such as calcium that may enter the pipette, bind to the dyes, and change their effective polarity. So in general, dyes and small molecules are easy to deliver into cells using short duration pulses or short trains. We like to use high voltage and high concentrations for delivery of a rapid high bolus to a single cell. DNA, of course, is larger and takes longer trains. It'd be nice to combine these two and label cells with dye and DNA to look at a cell morphology before the DNA is expressed, but by the time you get DNA into one cell, typically you've labeled multiple cells with dye, so that's usually doesn't work out very well. Finally, I'll just talk about troubleshooting common problems. So the most common problem is not seeing any DNA, any protein expression following single-cell electroporation of DNA. There are many steps that you have to do at this point. You have to check the quality of your DNA and its concentration. You have to make sure that the tip geometries are optimal. And this is very challenging to do with DNA since there's usually a 24-hour delay before you're testing. You see the protein expression, so it's very frustrating going back and forth. And the best thing to do at that point is to try single-cell electroporation of fluorescent dyes, which give you an immediate feedback. If that works, those pipettes should be good for DNA as well. Of course, you have to make sure you're using the correct polarity, and you always want to check your circuitry and make sure that your ground electrode is connected properly. If you fail to label cells with dye by single-cell electroporation, you have to, again, make sure that your tip geometry is optimal. That's usually a common problem that you have to adjust. It's the same thing. You have to make sure that you're using the correct polarity and your ground electrode is correct. Always try to use fresh dye. And you can directly visualize the movement of the dye with fluorescent microscopy. If you're using the wrong polarity, you'll see dye moving up into the pipette away from the tip. If it's the correct polarity, you'll see a dye moving ionizing outside the pipette. But again, don't overexcite with fluorescence because that will damage your cells. Lastly, clogging the tip can be very problematic. To deal with this, you can spin down your solutions before loading. If you're getting multiple cells labeled with dye or DNA, simply decrease tip diameters, shorten the duration of your stimulation, and reduce the voltage. And again, that's something that's very easy to do with the Axoporator 800A. For single-cell electroporation of dye, if you're only getting dim cells, you can increase the pulse or train duration, increase your dye concentration, make sure you're using fresh dye stocks. And if your labeled cells are fading and disappearing, it's often due to people using fluorescence while they're labeling. Typically, we have the fluorescence on to visualize loading of cells to optimize tip parameters and geometries and stimulation parameters, and then we turn that to fluorescence off and use those same parameters under a blind fashion to label cells, and then quickly check the fluorescence maybe minutes later when the dye fills the cells to see if we have loading. But we're very careful not to overexpose those cells because they will die and fade and disappear. So these are references for this talk. We have our methods on both a Jove protocol as well as there's our original paper in Neuron from 2001. So I think with that, I'll hand this over to Jeffrey for questions.
All right. Wow. Thank you so much. It is a wonderful presentation. I learned a lot. So for those attendees, if you have any question right now, you can type it in the Q&A sections. Hi, Kurt. I have a couple questions to ask you.
Okay.
Yeah, the first question is that so you talk about a lot about how to make a optimize, how to do the optimized electroporation, right? So in the technical point is that the pipette is physically touched to the cell, or it is just not touching to the cell when I do the electroporation, and how do we know what is the optimal working distance, for example, for target SCE, single cell electroporation, right? Basically, right?
Yeah. So the tip has to be either very, very close to the cell or touching it. I think optimally it touches the cell.
But how do we know it's touching to the cell? How do we know?
Well, if you're doing blind single-cell electroporation, you don't know, and that's where you just have to do many different repetitions of stimulations. But if you're using targeted, you can actually see it touch the cell. You definitely don't need to make a very strong patch. You don't need a gigohm seal at all. And there's also a problem if you stimulate with too high of a voltage, you actually can adhere the tip to the cell, and that may be a problem when you're actually removing the pipette. So often, if you're doing targeted, you can see those events happen and try to adjust your stimulation parameters, decreasing the voltage, typically to prevent that sort of fusion event from happening. Another approach to helping that is to quickly extract the pipette, which usually, if there is fusion, help the cell just reseal very quickly.
I see. All right. So the second question about what is the optimal pipette resistance that you use? Is that depends on what kind of cell you electroporated, or depends on what kind of diet or what kind of material that you are going to electroporate to the cell? I mean, what is the optimal pipette resistance?
So the resistance will be both a feature of the tip diameter of the pipette as well as whatever the solution is that you're using inside the pipette. So that will vary depending on your solutions. For us in our preparations, a 30 megaohm resistance pipette reliably works well for us. There might be some difference for different solutions that people have.
I see. And one of the questions is that you mentioned that you did it in the slide culture for the hippocampus neurons. How about, have you done anything in the brain slide preparation?
No, I've only done in organotypic slices. Other groups have electroporated mouse neurons, both in acute slices and in vivo in cortex. And using targeted electroporation significantly improves their success. So having correct tip cell geometries, contacts is probably very important for making that happen. Other groups who have tried doing blind single-cell electroporation in mammalian systems have had very low or lower efficiency.
Okay. The last question that I have is that, right now it's very hard for optogenetics, right? Has you or any group tried to, for example, to inject like channel rhodopsins expressing DNA to the neurons, or acute slice, or culture, or in vivo using Axoporator, right?
Yeah, so we've been very interested in trying the channel rhodopsin, halorhodopsin techniques. Unfortunately, since we're dealing in our lab, we deal with a visual circuit. It's a little challenging to use a tool that requires a visual spectrum light stimulation. But what we have used are dreads. So these are channels that are you can activate with an exogenous drug that only activates those channels, and they're very effective for increasing or decreasing firing rates. And they work very well electroporated into cells and then driven by external drugs. So I think a lot of these tools are absolutely, there's no reason why they wouldn't be very useful with single-cell electroporation.
All right, thank you. Hi, Kurt, can you hand the ball to me?
Sure.
Right.
Give me a second.
And I would like to thank you for your attending, and we look forward to seeing you at future events right here at marketer.webex.com. Have a wonderful day. Thank you.